This video is going to aim to clarify the principles that go into quantitative genetics, specifically dealing with the variances that we describe in the main lecture. We'll start by having just some overview of what we mean by phenotypic variance, and then we'll drill into the other subforms of variances and end with some idea of heritability and what heritability tells us and what it doesn't tell us and all of that. So our first real key into quantitative genetics really relies on understanding what we mean by phenotypic variance and what the aim of quantitative genetics really is. So what we mean by phenotypic variance is that most complex traits, and by complex traits, I really mean traits that are controlled by many different factors. That can be many different genes or genes and environmental factors. Those traits vary in their phenotype along a spectrum. So I have here a bell curve, which really represents what we mean in general. For any trait, we have very, very few individuals at one extreme of the phenotype, very few individuals at the other extreme of the phenotype, and as we get closer and closer and closer to the average, we see more and more individuals lying towards that average. This gets to traditional statistics ideas such as standard deviations from the mean and all of that, but most complex multifactorial traits, most traits that are responding to multiple factors, such as multiple genes, follow this general idea. And although I used the example in the lecture, and I hate to reuse an example, I really think that height in humans is the perfect example of such a trait, because there is the smallest person who ever lived on record, and there is the tallest person who ever lived on record. But those single smallest and single tallest people, they represent by definition the ends of the spectrum, the absolute extremes. And now we can have short people, very short people, but they're not entirely rare. We know short people. And we can have tall people, the kind of people where we look on the street and say, my goodness, look how tall that guy is. I mean, that's not extremely rare. That's possible. But we expect to see, and indeed our personal experience is that most individuals lie close to the average height. That's why we think of it as the average height. It is the height most commonly seen. All of this spectrum, however, all of this variation from one extreme of a phenotype to the other is what we mean by the phenotypic variance. The phenotypic variance can be thought of as how a single trait and all of the quantitative phenotypes of that trait vary across a spectrum from one extreme to the other. The phenotypic variance is how much we are different from one another for a particular trait. Now, quantitative genetics aims to explain that variance. Quantitative genetics aims to measure, understand, and explain what it is that makes us different for a particular phenotype, for a particular trait. And I have found in my experience that the best way, I think, to understand this is through a pie chart, where the entire pie represents 100% of the components that feed into phenotypic variance. Now again, let me put that back into context. What I mean by that is that the entirety of this pie chart explains 100% of why individuals are different from each other across an entire spectrum for a particular trait. When we are done filling in this pie chart, we will have an example, perhaps, of all of the different things, all of the different differences that exist between individuals to explain the differences we see in their height. So let's consider the first wedge of this pie chart. What is one way you can think of that would explain why people differ in their height? I'm hoping some of you said first, well, the environment. Environmental effects can indeed lead to differences in height. Malnutrition can cause someone to grow quite short. An abundance of nutrition can allow a person to reach the full potential of their height. So changes in an individual's environment can be responsible for differences in individuals' height. Better nutrition, more vitamins, better food can allow someone to grow taller. worse nutrition, worse food, less vitamins can force a person to grow shorter. So the environment plays a role in the differences we see between people for this trait. And that variation in environment explains a piece of the variations we see across individuals for their phenotypes. And we call those variations in environment the environmental variance. Now we can give this environmental variance an arbitrary number, such as 0.15. And what we mean there is that 15% of the differences that we see between individuals for height are due to the differences in their environment. If 15% of the differences we see in individuals is due to differences in their environment, what is the other 85% due to? Hopefully you said that the other 85% is due to differences in individuals' genes. We call that the genetic variance. Again, to summarize where we are so far, what we are saying is that people differ from one another in height, and that represents the phenotypic variance. And then we ask as quantitative geneticists, why do people vary in their height? Well, we can say that 15% of the variation in height is due to variations in environment. Some people eat better, some people eat worse. Some people take vitamins, some people don't. The other 85% of the variation we see across individuals for height is due to differences in their genes. But when it comes to that genetic variance, that sum total of all the differences in genes, we can parse that out into individual variances as well. We can break the genetic variance up into 3 different categories. And those different categories represent the different gene classes we already know of. The first gene class is the traditional Mendelian class. Mendel based his entire understanding on genetics in the principle that is represented here in this simple schematic. Mendel's theory of genetics and inheritance hinged on the concept of dominance. Mendel said that any individual with no dominant alleles will have a phenotype of one type. As soon as a dominant allele was introduced, that phenotype would switch to the dominant phenotype. From that point on, it did not matter how many dominant alleles there were. In other words, Mendelian genetics states that whether you are homozygotic dominant or heterozygotic, you will have the same exact phenotype. The only difference in phenotype comes from being recessive. That's Mendelian genetics. And again, it hinges on the concept of dominance. So genes that behave in this way can affect the genetic variance that we see for a particular trait. And we refer to that variance as dominance genetic variance, where again, that term dominance reminds us that we're dealing with Mendelian genes, which hinge on dominance. So again, the dominant genetic variance of a trait is the component of that trait that is due to genes that behave in a Mendelian way. If we gave the dominant genetic variance a value of 0.2, what we are saying is that 20% of the cause for why individuals differ in their phenotype for a particular trait is due to differences in genes that behave in a Mendelian way. The second class of genes that we're aware of are genes where, yes, the dominant, homozygous dominant, and heterozygous genotypes behave differently than the recessive genotype. But we can't predict anything about those genes unless we know something about the gene that is controlling them. And that key point where we're talking about gene control should remind you very quickly of epistasis. Epistatic genes are not Mendelian genes. They do not behave in the same Mendelian fashion. We cannot predict the phenotype of epistatic genes by knowing the genotype alone. We need to know something about the relationship and the control of one gene to another. So epistatic genes and their effect on the phenotypic variance need to be considered separately. And we call that variance the Genic interaction variance. That is, we refer to these genes as interacting genes, where one gene controls another. And the Genic interaction variance in this example, we can say is 0.35. What that number tells us is that 35% of the reason for why different individuals differ from one another for a particular trait and a particular phenotype is due to differences they have in their epistatic genes. The last class of genes that we really didn't touch on until this lecture are genes that behave in a unique way. These are genes where the phenotype of the homozygous recessive individual is the least abundant or least robust, the least extreme of the phenotypes possible. The presence of one dominant allele grants some of the dominant phenotype, and the presence of two dominant alleles grants even more of that dominant phenotype. Again, if we think of height, the homozygous recessive individual would be the shortest, the heterozygote would be taller, and the homozygote dominant would be taller still. It's almost as if the effect of the dominant allele sums up across the genotype. Because of this idea of the allele effects summing up, we refer to genes of this type as additive genes. And they make up the last class of the phenotypic variance quantitative genetic story. The additive genetic variance is the proportion of the phenotypic variance due to differences in genes that behave in this additive way. While I've been making these numbers up all along, I can fill in the additive variance with a great deal of certainty. In this example, the additive genetic variance would be 0.3, because it completes our pie chart. If you remember when I started this review video, I said that we are trying to explain 100% of the phenotypic variance. We're trying to account for all of the factors that cause people to be different in their height across the entire spectrum of that phenotype. And what we have done in this example is said that 15% of those differences are due to differences in individuals' environments. 20% of those differences in height are due to differences in the genes of people that behave in a Mendelian way. 35% of the differences we see in height are due to differences that individuals have in their epistatic genes, and 30% of the differences we see for that phenotype are due to differences that individuals have in the genes that behave additively. Taken together, we have explained 100% of the phenotypic variance in this example. If we add up each of these individual components, we'll see that it totals 1. And again, if we go back to the idea of overall genetic variance that we started with before, we had said that the genetic variance in this case would be 0.85. And indeed, if we take the 30% of the additive genes, the 35% of the epistatic genes, and the 20% of the Mendelian genes, we see that all of the genetic components together total 85% or 0.85. We'll end this video by distinguishing between two types of heritability, broad sense and narrow sense. Quantitative genetics arose, at least in part, due to breeding programs and artificial selection. Farmers wanting fatter chickens and more milk-producing cows, people that grow crops wanting higher yielding crops, more drought-tolerant crops. And what all of these individuals have in common is that they appreciate the value of and often engage in breeding programs. Breeding programs are artificial selection programs where desirable traits are bred for or selected for, and undesirable traits are bred out or selected against. So imagine the simplest case. Imagine a scenario where for a trait, the environmental variance was 0.9. What does that value mean? Well, that means that 90% of the phenotypic variance, that is 90% of the differences we see between individuals for a trait, is due to differences in their environment. If we use the example of egg laying in chickens, if egg laying had an environmental variance of 0.9, there would be no need to breed for egg-laying traits. If you wanted all of your chickens to lay a lot of eggs, and the environmental variance was 0.9, you, as a farmer, you should pay attention to where are the high egg-laying chickens sleeping. Get all of your chickens to sleep there. What feed are the high egg-laying chickens eating? Feed that feed to all of your chickens. Where do your high egg-laying chickens go in the afternoon? Roust all of your chickens to that area. In other words, if the environmental variance is 0.9, that means 90% of the differences you see in your chickens are due to environmental differences. If you want all of the chickens to lay the same amount of eggs, and 90% of that trait is controlled by the environment, then just mimic the environment of the high egg layers for all of your chickens. But most traits don't behave that way. Most traits have a strong genetic component. So if we had an environmental variance of 0.1, we would know that 10% of the differences we see in individuals is due to differences in their environment, but the remaining 90% would make up the genetic variance. And there, if we want to breed a desirable trait, we have to literally go to breeding programs to do it. When we are taking a broad perspective on breeding programs, we call that a broad sense heritability. We look at all of the genetic factors for a trait. We look at the dominant genetic variance, the genic interaction variance, and the additive genetic variance. The broad sense heritability takes the broadest look at a gene's control. Now, you can probably appreciate the fact that from a breeder's perspective, some of these genes are easier to track than others. For dominant genetic variance, when a farmer sees that one of their animals is showing the dominant trait, there could be confusion there, because both of these genotypes lead to a dominant phenotype. Farmers, breeders, don't like ambiguity, and they don't like guessing the genotype. So dominant genetic variance is less than ideal for breeders, because they can't distinguish between a homozygotic dominant individual and a heterozygote. Even more so, for epistasis, there's even more ambiguity, because the effects of one gene can be completely masked by the genotype of another, whereas that second gene can have the same exact genotype, but we only see it when we're allowed to view the expression of that gene by the epistatic gene that controls it. Epistatic genes lead to a great deal of confusion in breeding programs because epistatic genes can often be masked, making it impossible to see the genotype in the livestock or the plants that you're trying to raise. But additive genes provide a what you see is what you get perspective of phenotype. A short plant is unambiguously homozygotic recessive. A mid-sized plant is unambiguously heterozygotic, and a tall plant is definitely homozygotic dominant for additive genes. Because the effects of individual alleles are observable only for additive genes, phenotypes and genotypes are often closely matched for this gene type. For that reason, breeders love additive genes, because they can very precisely view and determine the genotype of their animals, and then breed animals that they want to have bred based on those genotypes. Because additive genes are so important for breeders, those breeders that want to specifically focus their breeding program on the genetic component of a trait will often narrowly focus on additive genes. And so when we talk about a narrow sense heritability, we are referring to a heritability program, a breeding program, that is focusing exclusively on the additive genetic variance for a trait, and disregarding the other genetic variances simply because they're too confusing to determine and they lead to too much ambiguity in the breeding program. So broad sense heritability takes all different types of genetic variances into account, whereas the narrow sense heritability focuses exclusively on additive genetic variance.